Hexactinellida, the glass sponges. Of 7,000 species of sponges, about 600 belong to the class of so-called glass sponges, or sixth-rate sponges, better known as Hexactinellida. Glass sponges are exclusively marine animals that are found at depths ranging from 10 meters to 7,000 meters in all oceans. The vast majority of sponges live at depths of 200 to 2,000 meters, though. They live in cold temperate waters with a temperature usually between 2 to 11 degrees Celsius. This leaves a majority of sponges living in deep, cold waters farther from land. Some sponges do live in reefs, though.
Fossils suggest that glass sponges were established by the late Proterozoic era. In the Jurassic, there were large reefs formed of glass sponges that covered the northern shore of the Tethys Sea, which is now the area representing Europe and eastern Canada. Sponge reefs, as biogenic structure, were initially thought to be extinct until the discovery of the reefs in the Pacific Northwest. Most glass sponges live in depths greater than 500 meters. Off the coast of British Columbia, in the North Pacific, vast reefs spanning hundreds of square kilometers live in shallower depths of approximately 200 meters. This depth is speculated to be favorable for sponge reefs because of high silica content, high food content, high water flow, and cold temperatures reminiscent of the deep sea.
Their size can vary from a few centimeters to several meters. Under extreme circumstances, those certain glass sponges, more specifically, the Antarctic glass sponge, Rosella nuda, and Scolimastra jobini, can weigh up to 600 pounds and grow sometimes to 2 meters high. Another remarkable property of glass sponges is their particularly long survival. Hexactinellida are champions among multicellular animals in longevity. An age of 500 is common for them, with extreme sponges living in what is believed to be 1,500 years. This is due to the fact that they live in extremely cold waters, which slows their metabolic rate, as well as the fact that there are no real predators to glass sponges as they are made of their silicon spiracles. With 5 orders, 17 families, and 118 genera, there is a lot to know about the class Hexactinellida. Basic hexactinellids are radially symmetrical and have various modifications made to their cylindrical tubular construction. The ultra-thin organization of spicules in many sixth-rate sponges is a system of thin layers deposited around a central cylinder, which includes inside a square channel filled with organic matter. This is what possibly gives sponges the look that they are a combination of multiple sponges. Coloration in most is pale or light yellow. Much of the sponge's body is composed of syncytial tissue and extensive regions of multinucleate cytoplasm. In particular, the epidermal cells of other sponges are absent, being replaced by a syncytial net of amoebocytes, through which the spicules penetrate. Unlike other sponges, they do not possess the ability to contract. Sixth-rate sponges are peculiar in having an amazing variety of spicules. More than 400 terms have been proposed for the taxonomy of hexactinellids to describe the morphology of the spicules. First, all the spicules are divided into so-called macrosclers and microsclers. These definitions are rather traditional because microsclers are sometimes bigger than some types of macrosclers. Nevertheless, macrosclers are usually the biggest spicules with the function of maintaining the animal's body. The role of microsclers and their functional significance are still unknown, though they are important for species identification.
Glass sponges possess a unique silicon dioxide skeleton and syncytial tissue formed by fused embryonic cells. They're typically vase, plate, or tube-shaped. There are 2 structural types of glass sponges. Lysocene sponges have a loose spicule skeleton and are non-reef-forming. Dictyonine, on the other hand, can form reefs due to a fused spicule skeleton.
For feeding, hexactinellids possess a cavernous central cavity, the atrium, through which water passes. By passing water through their central cavity, glass sponges are able to filter food from the water. A few species of sponges even form symbiotic relationships with endosymbiotes. A cool feature developed by a few species involves the silicone spicule's ability to transmit photons from the surface of spicules into the body of a glass sponge. The light is passed through the spicules from the surface to deeper layers, and it is assumed that the photons generated by triboluminescence can be used by symbiotic bacteria that are localized on the surface of the spicules of hexactinellids. Adapted to life in absolute darkness, these sponges have light-transmitting spicules that are capable of generating electromagnetic radiation, in particular in the visual spectrum, allowing some to support symbiotic photosynthesizing cyanobacteria, even though they live at oceanic depths that are inaccessible to sunlight photons. Sperm are released into the water and drift before they are being taken into an organism with water. They can make their way to the eggs within the organism. After fertilization, the larvae are incubated for a relatively long time, so long that they even form spicules before being released as larvae. These differ from other sponge larvae in lacking flagella or any other method of locomotion. Therefore, hexactinellids do not drift too far before setting. After a larva lands on the ocean floor, it metamorphoses and the adult sponge begins to grow. Euplactella, commonly known as Venus's flower basket because of its obvious appearance,